In this video, we learn about the Krebs cycle, also known as the citric acid cycle. During glycolysis, glucose is converted through a series of reactions into pyruvate, which is further processed in the second step in glucose oxidation. This step is catalyzed by the enzyme pyruvate dehydrogenase and takes place in the mitochondrial matrix. In the presence of oxygen, pyruvate gets converted into acetyl coenzyme A, and during this process, another molecule of NADH is synthesized, and one of the carbon atoms in pyruvate is oxidized to carbon dioxide. The pyruvate formed at the end of glycolysis is converted to acetyl coenzyme A. The processing of pyruvate is controlled both by positive control and negative control. When ATP is abundant, the enzyme complex that catalyzes the conversion of pyruvate to acetyl coenzyme A is inhibited. On the other hand, the enzyme complex is stimulated in the absence of ATP. Once acetyl coenzyme A is formed, it enters the citric acid cycle, which is the third step of glucose oxidation. The series of reactions that are part of the citric acid cycle take place in the mitochondrial matrix. Each acetyl coenzyme A molecule is oxidized to two molecules of carbon dioxide. Some of the potential energy that is released during this process is used to reduce NAD+ to NADH, which is a high-energy intermediate, reduce flavin adenine dinucleotide, or FAD, to FADH2, which is another high-energy intermediate, and to phosphorylate GDP to form GTP, which is later converted to ATP. Throughout the citric acid cycle, a series of carboxylic acids are oxidized and recycled. Citrate, the first molecule in the cycle, is formed from a reaction between acetyl coenzyme A and oxaloacetate. So the two carbons in acetyl coenzyme A enter the citric acid cycle by being added onto the three carbons with an oxaloacetate, forming citrate. The citric acid cycle completes glucose oxidation. The energy released by the oxidation of one acetyl coenzyme A molecule is used to produce three NADH molecules, one FADH2 molecule, and one GTP molecule. The GTP molecule is later converted to an ATP molecule. The carbons, shown in blue in citrate, are subsequently released as carbon dioxide. So we start with a 6-carbon molecule and end up with a 4-carbon molecule. Feedback inhibition plays an important role in regulating the citric acid cycle. This cycle can be turned off at multiple points. It starts with acetyl coenzyme A and ends with carbon dioxide. The potential energy that is released throughout the cycle is used to produce NADH and FADH2, which are high-energy intermediates, also known as electron carriers, and ATP. When the energy supply is high, meaning that there are a lot of ATP molecules around in the cell, the cycle slows down, and when ATP is scarce, the cycle is stimulated. A summary of how the citric acid cycle is regulated is shown on this slide. At the end of glycolysis, pyruvate oxidation and the citric acid cycle One molecule of glucose is completely oxidized to carbon dioxide. Glucose oxidation also leads to the production of NADH, FADH2, and ATP. If you look at an energy diagram for the process of glucose oxidation, where free energy change relative to glucose is depicted on the y-axis, and the oxidation of glucose is represented on the x-axis, we can see glucose at 0 kilocalories per mole because none of the energy contained within glucose has yet been released. Each molecule of glucose that is oxidized reduces 10 molecules of NAD to NADH. So we have 2 plus 2 plus 2 plus 2 plus 2 equals 10. 2 molecules of FAD are reduced to FADH2 and 4 molecules of ATP are produced through substrate level phosphorylation. Most of the original energy in glucose is now contained in the electrons transferred to NADH and FADH2. These electrons are ultimately transferred to oxygen, which is the final electron acceptor. And the energy released during glucose oxidation, which is contained within NADH and FADH2, will be used later to synthesize ATP during oxidative phosphorylation. This brings us to the end of the discussion about Krebs cycle. After going through this video, I'd like you to be able to respond to the questions listed on this slide. Thank you.